Because we have a packed agenda, I do think we can get started. So as I said, thank you so much for joining us today for the next PanKbase webinar and office hours. I'm going to share some updates from PanKbase itself, go through some feedback, and then we'll go through some tutorials and Q&A from our featured speakers. So first off, I just want to remind everyone that we have PanKbase on our various social medias. So you can join us on X, Blue Sky, and LinkedIn. So you have different groups where you can join us for that. This is where you can get all of our information in terms of latest production release items, but also any of the events that we have ongoing. And I just want to remind everyone that our next upcoming webinar is going to be October 20th. So please keep that on your radar. If you are on this webinar right now, you do not need to worry about registering again. So we hope to see you there. And those are my updates for today. And so we'll get into our featured speakers. So I am going to share screen and then we can have Anna, who is going to kick us off first. So give me 1 second. Okay.
Super. Thank you, Mackenzie. Well, good morning, everyone from North California. It's a pleasure to be here today and to have the opportunity to represent the PanKbase team and to give you an update on our platform. We're going to tell you today about powering your next discovery and introduce you to some new data and tools that are available in PanKbase. I'm just one of 4 members of our team who will be sharing updates with you this morning. I'm Anna Gloyn and I'm based at Stanford University. Next slide. So just to set the stage, what you can expect today is we're going to give you a very brief reminder of who PanKbase are, in case this is your first rodeo and you've not heard about us before. And then we're going to tell you about some new developments in the browser with a focus on functional data. Tom, my colleague from Vanderbilt, will do a deep dive on the types of data that we've integrated. And then Annie is going to give you a demo on how you can explore those data on the PanKbase website. And then we're going to wrap up with Ringo from Michigan, who's going to share with you some of the developments that have been made with natural language models and give you a demo, I believe, on how that all works together. So we think it might work best to take questions after Tom and I have spoken and maybe before Annie gives you the demo. But if you've got burning questions, please feel free to put them in the chat and we will try and respond in the most efficient manner. Throughout the talk, you can see some QR codes. These will take you to the websites that we're talking about. So please feel free to take screenshots. And of course, the slides and recording will be available afterwards. All right, I think we're ready to press play, Mackenzie. Okay, so a reminder of PanKbase, who and what we are. We're a relatively new initiative funded by NIDDK and part of the Human Islet Research Network, who've been funded to develop a platform to provide data, knowledge, and analytical tools on the human pancreas with a view to facilitating research in type 1 diabetes using modern data science tools and analyses. As you can see from the mugshots here on the screen, we are a large interdisciplinary team across multiple institutions. We have expertise in platform development, type 1 diabetes, data science, computational biology, islet biology, as well as engagement and outreach. And we work together with our extended and amazing teams to really deliver what you're going to hear about today. Next slide. So over the last year and at a number of similar events to these, we've shared with you the integrated single cell browser that we've put together. This was one of the tools that was in high demand. There was an urgent need to assemble and integrate data on human islets from multiple studies. You can continue to explore our data here at the QR code. You can read about the paper that's available on bioRxiv. But of course, this is just one type of data that's been assembled by the amazing Islet community. There are other types of data that we would like to integrate. I'm not going to have time today to go into some of the other related browsers, but please think about taking a moment to explore our genome browser, differential gene expression browser, and some of our other tools that are available on PanKbase. But today, what we really want to do is feature something which was an ask from the community. Next slide, please, Mackenzie. So, over the course of the last year, at multiple outreach events at the American Diabetes Association meetings through HERN and through Breakthrough T1D, we've heard consistently that there is an interest from the community to have access to functional data. And we've been trying to grapple with the many challenges that we face when we try to bring data together from multiple sources. So many of you on the call will not be necessarily experts in how you generate functional data, but fear not, we have Tom with us today who's going to give you a deep dive in a moment into how functional data can be generated. But the key things to know are that there are many different methods and protocols that are deployed in labs around the world to study the function of islets. On top of that, there are also many different sources of human islets. We can have them from cadaveric donors, we can have living donors, we can have isolated islets, or even pancreas slices. So when we start to think about this complexity, there are lots of things we have to consider when we want to start to aggregate functional data and make it available to the community. So firstly, we've got to think about the source of the islets. Secondly, we've got to consider the different methods and protocols. Thirdly, we've got to think about all the different variables that will influence the results. And then finally, we've got to think about the fact that the data outputs could be very different. And essentially what this means is there's no one easy fit to bring this data together. But we're very fortunate because among our community, there are large data sets that are available to us, which have been prepared in a more homogeneous fashion. And this allows us to avoid some of these challenges. So in the PanKbase community, we took the decision to focus on one of these large resources, the Integrated Islet Distribution Program, IIDP, which I'm sure is known to many of you because they distribute islets all around the world to islet biologists like yourselves to use in your own studies. But there's a huge advantage to starting with that data set. And I'm going to hand over now to Tom, who's going to set up the stage for the data that's been taken and put into PanKbase, and then Annie's going to show you how it can be used. Over to you, Tom.
All right, thank you, Anna. Yeah, so as Anna introduced, we're going to be talking about data that was developed through the Integrated Islet Distribution Program. And on the screen here are the sort of the multiple PIs that are associated with this program. And so we have several sort of sub-programs under the IDP, and what we'll talk about today is the Human Islet Phenotyping Program, HIP, and the Human Islet Genotyping Initiative, which is HIGI. Next slide, please. So the goals of these programs are to essentially integrate multimodal data across different cohorts, and essentially we've a sort of centralized phenotyping and genotyping program has been conducted to study function and across a diverse cohort of donors. Next slide, please. So when we talk about functional data, the Human Islet Phenotyping Program has been running the same protocol on hundreds of human islet donor preps. And so what we're really talking about when we talk about functional data in the context of HIP is that islets are perfused with media in chambers. And so we have this dynamic secretion profile. Next slide, please. And can you go on ahead? Yeah, thank you. So what do we get from this platform? We get insulin and glucagon secretion profiles over time with sequential secretagogues. The baseline is 5.6 millimolar glucose. And so you can see at the top of these graphs, the secretagogues are labeled. And so that 5.6 is our physiological baseline. And then there's high glucose, which is 16.7 millimolar glucose. And then we have a high glucose with IBMX condition, which stimulates glucagon, insulin and insulin secretion via cyclic AMP potentiation. And then we have a low glucose, which is G1.7 millimolar, plus adrenaline or epinephrine. And this is to simulate the hypoglycemia response. And then finally, we have potassium chloride, which depolarizes the membrane and drives secretion as well. And so we have these in these sequential secretagogues, which are run by the islets, and we measure both insulin and glucagon. And so with this data, we can integrate the area under the curves and get magnitude of secretion under different secretagogues. And you can imagine that there's plenty of other analyses that could be run, if we think about time dependence and these sorts of things. So next slide, please. So alongside the functional data, we also collect imaging data with which we can analyze the composition of the human islets from the same preparations. Can we go one ahead? And so we can essentially start drawing relationships between islet composition and other donor metadata such as sex and genetic ancestry, if we go one ahead. Um, and next slide, please, as well. Moreover, we can, draw associations between secretion traits and islet composition, as well as hormone content as well. And so this is work that was published recently by the team in Nature Communications, showing that there are strong relationships, in particular between insulin secretion and islet cell type composition. Can we go on ahead, please? And also glucagon secretion as well, but particularly content in this case. Um, let's go one ahead. And also with the with work done by the Human Islet Genotyping Initiative as well, we were able to draw correlations between diabetes risk scores and islet features. So these would be the secretion traits as well as cell composition. And yeah, so we saw that there was a significant relationship in here in particular between the delta cell composition and the type 2 diabetes genetic risk score as well. So can we go one ahead? Yeah, so that's just pointing those out. So this is just to give a feel of, what can be done with this kind of data and the potential for integration across modalities. So yeah, we've seen that islet composition strongly associates with both secretion traits as well as genetically predicted ancestry, and um, so it raises the question whether like islet heterogeneity in terms of composition might underlie heterogeneity that is observed in diabetes. Um, yeah, as we saw, delta cells were associated with risk score for type 2 diabetes. Um, and yeah, this could have implications, for beta cell replacement therapy as well as like the pathophysiology of diabetes. So um, Yeah, so just to acknowledge the teams, this is a huge team effort, and this is the Human Islet Phenotyping Program. Yeah, we there's a lot of people involved. I'd like to highlight especially Anastasia and May, who are tirelessly uh, getting this data, the functional data together. Weekly shipments, and yeah, really great effort on their part. Next. And then also the Human Islet Genotyping Initiative, and there's been a huge effort in genotyping these donors, so yeah, that's predominantly Anna Gloyn's group at Stanford. So and
Fant-
next,
Fantastic.
that's
I think,
cool.
yeah. So thank you, Tom. That's fantastic. And the opportunity now is these data that Tom's just walked you through have been integrated into the PanKbase platform. And we've got Annie, one of our developers, on the call, and she's going to give you a live demo of how you can play with this data, how you can explore it. And after she's done that, Tom and I and Annie would be happy to take any questions that people on the call have around how you might use this data. And then we can tell you some of the things that are on the tables for what we would like to do next with other types of data in the functional realm that we hope to integrate. Annie, over to you for a demo.
Hi. Can you guys all hear and see me?
Yes, we can hear and see.
Awesome, thank you. So, what I'm going to do is I'm just going to walk you through in real time how you can explore the functional data that are already made available up on this PanKbase website. This is pankbase.org. You can follow along if you want. You can play with it in however whatever way you see fit. But we're just going to go here under the Resources tab to this functional browser. And what we have is 413 donors as a subset of this greater pool of donors, but 413 of them have these available functional data. We have two assays. One is an islet insulin secretion assay, one is an islet glucagon secretion assay. So in terms of how the data was collected, I think the scientists are better poised to speak to that than I am. But what we have here basically is an interactive explorer tool that you can use that responds in real time to the filters you impose. So we can say, for instance, we want to view these insulin data, and let's restrict the age. Let's say only donors above 50 will, we can just make it round, above 50. So it looks like we have 148 donors that meet these criteria. You can toggle on and off viewing the entire donor pool versus just the filtered donors. So you'll see, whoops. Yeah, it does preserve. Uh, and then you can also, if you want, view individual donor traces, and you can do things like, oh, what is this one donor who has in the potassium chloride who has this high secretion, and this is one particular donor, 59-year-old male with this particular accession number, if you wanted to view that, that donor's data individually. And you can also see that the filters that you impose are reflected in, if you go down here, these results, which are in a sortable table. You can sort, by BMI, you can sort by age, and this is already, again, this filtered pool that is 50 and up. You can download the data into, whatever file format that you want. And it gets, you can get very granular. You can say, okay, I only want to view certain isolation centers, and you can filter the like peptide um, values and things like that. You might get a very small pool. Okay, now we're down to 55. But you can broaden that pool back out. Uh, and this is really just an interactive uh it's like you're looking through peeling back all these layers in real time, which I think is kind of neat. You can also toggle it to the percentage content. Um, Again, these are just different ways of viewing and visualizing the same data. You can, at all times, you can always see this all, without having to broaden the entire filter pool back out. You can see the entire donor pool all at one go, and it will still show you, this is a 7-year-old, who had this very high uh, peak here. Um, and again, you can always view the individual donors that are filtered for your specific criteria. Now we can also go here and view some summary status. So we can say, for instance, why are we only getting the control without diabetes? Yeah, okay. So whatever traits are present in the pool that you have filtered, and we can let's broaden it all the way back out by showing all the isolation centers. Let's do all. Yeah, here we go. Yeah, if you're seeing missing traits, it might just be because you filtered the data down a little bit too far. But we have these derived quantities that are calculated from HbA1c. And again, I don't know the threshold that is used to determine that, but somebody does. You can view, um, the secretion under all these different conditions sequentially through the course of the assay by, like, are they pre-diabetic, non-diabetic, or are they diabetic? And you can view it by reported gender. You can view, any trait. I think there's an ethnicity tab. You can view according to which isolation center these samples were collected at. And this is, again, as you sort of read through these conditions, these are the sequential, like every phase of the assay gets its own little chart. And you can also view functional trait associations. These are pre-calculated. I believe these are just already calculated, and they're not dynamically done on the fly, but these are some calculations that we already have. According to the functional traits that we have available in the collected data. But I think the perifusion traces are the most exciting part. I think that's kind of what everybody maybe is here to see. Maybe not. Maybe everybody's here. And, I've been using the insulin tab to kind of show you that, but we also do have glucagon, and, people are going to have, certain outstanding, very high outlier donors that you can mouse over and view that. Oh, and the other thing that I guess we can do is if you already know which set of donors you would like to, a set of individual donors that you would like to look at, let's hide these filters. You can do something like copying the accession number, and if you come directly to the PanKbase page and you say, oh, I want to see that donor that I was interested in, you can go straight to select donors, paste in the accession number, and it will give you the one individual donor what? I don't know why it isn't showing up. It'll certainly show it in I think that might be an issue that we'll have to address.
Some donors, especially type 1 diabetes donors, because they are T1D, so there is basically no beta cell left. It's normal to have some donor have zero functional data, and
Okay.
that's what it is. Yeah.
Let's see if we get, yeah, here we go. Thank you. Okay, I was worried there for a second. So if we have individual donors, yeah, it looks like we only have that one who does not have a diabetes flag in the. Thank you, Ringo. But yeah, so you can really, we've made it very easy to kind of build your own filtered view of these results. And one last thing that I want to show you that is interacts with some beautiful work done by Alex Schillen, our a front-end developer and designer, is that we have a a summary of all, yeah, the donor summary here. And what we can do is we can apply some filters. So let's say, under 45. And let's say low BMI. And okay, so we have this pool that we've created of 104 donors. And what we should be able to do is say visualize data in functional browser, and it will already, it will preserve those filters. And you can see that we've got a young donor pool with a lower BMI than this sort of median of this population. And you can see, again, we've got this 95% confidence interval that's visualized in red, but you can still mouse over and you can see all of these specific donors that were used to create this chart. That's pretty much what I've got for you today. Again, this is me as a developer showing a tool that I built. So if you have scientific insights or if you have things that you'd like a scientist perspective on, I think many people on this call are better placed than I am to address that. But if you have insights that you'd like to discuss with a developer, if you'd like to discuss the functionality of certain tools from a perspective that a developer can, I would love to hear about that.
Thank you so much, Annie. Really appreciate you taking us through the tool. Just out of interest, do we have a place where people can provide feedback? Because I expect there will be folks who will like to go away and have a play on the website themselves, explore the data. If they want to provide feedback, do we have an email they can reach or a place on the
I will drop that
website?
in the chat. There's a little contact page. And that shows you the ways that you can reach the PanKbase team. Yeah, we've
Perfect.
got
Perfect. Perfect. That is amazing. And I would encourage everybody on the call, if you're curious, go and have and explore. These things work best when you're kind of asking your own questions, playing around with the data. And then if you've got feedback, we would really love to hear that. Shall we pause, Mackenzie, for a moment? I don't see any questions, but if anybody has, now is your opportunity to ask the expert. Tom here, who I understand actually is at the bench and has done these assays. So if you have burning questions, he's here to ask and answer them.
Just a reminder that you can raise your hand. We can take you off of mute, and you can ask your questions that way. You can use the Q&A function at the top, or you can just put it into the chat and we can answer accordingly. It does look like there is a question in the Q&A. Albert has asked, can you talk about whether the PanKbase platform versus IIDP's own RDR platform have significant differences?
That is a great question, Albert. And if the others are comfortable, I might be the person best placed to answer that. So they do, yes. You're able to get far more information on the IIDP donors through the RDR from IIDP because there are certain data that are under an agreement with UNOS, which is the organ procurement regulators. And those data are only provided through IIDP and the RDR after you have made the commitments to treat those data with the respect they deserve. Also, at the moment on the RDR for IDP, you can look at an individual donor, but you can't work with the entire data set. So they're very complementary in terms of what they offer. And I think we have good communication. Both Marcella and myself are part of PanKbase and IIDP. So we're able to work with both teams to make sure that we're complementing effort and not duplicating. Albert, does that answer your question, or did you have something else that you were curious about that I didn't satisfy you with my answer with? I take the silence as satisfaction. Okay.
Thank you, Anna.
And then just really to whet your appetite, clearly the data set from IIDP is a phenomenal resource, but it is not the only show in town. There are lots of other data out there, some of which is done on the exact same platform at Vanderbilt. So there is the HPAP donors. So those can be integrated, and that allows us to extend our analysis from individual donors who are normoglycemic into the realms of people who lived with type 1 and type 2 diabetes. Um, Perifusion isn't the only way to measure islet function. We know that we have colleagues around the world, including up north in Canada, with the amazing effort that is led by Patrick McDonald at the Alberta Diabetes Institute, where they run static insulin secretion on all of their donors. Those data are also assembled and could be integrated into PanKbase. There will be challenges there. We're going to have to think about how you combine apples and pears. Perifusion and static secretion assays don't measure precisely the same thing. There are other perifusion protocols out there run by other labs, but they don't necessarily use the same protocol that the Vanderbilt pipeline does. So we'll have to get over some of those hurdles. We're thinking about integrations at different glucose concentrations and time points. And then Tom already whet your appetite with some of the beautiful phenotyping that's been done on cell composition. And at the moment, our cellular phenotyping is really restricted to looking at hormone response. But clearly, these other types of cellular phenotype can be integrated in to allow them to be used with other data. So that's where we're going, but these are really big tasks. It's why we're working not just within PanKbase, but with the broader Islet community, particularly our colleagues in the Human Islet Research Network, to really think about how we can tackle these important topics in a way that will best serve the community.
Yeah, there you go, Anna.
Thank you. What do you need to translate this data into the clinic for treatment? Zoe, that is a fantastic and incredibly important question. I'm just going to see if anyone else wants to take first stab at it before, just so you don't only hear my voice. Anybody else want to take first, give an opinion on that before I help out? Okay, I'm going to take that as me. So Zoe, I think we need to start with assembling the data to really understand what a healthy islet looks like. That's what we've done initially through the IIDP. Then we need to be able to understand how we can use that information to understand better what happens in people who've been living with type 1 and type 2 diabetes. And I think the next things that need to happen are to extend the types of analysis that we've done to clinical data. And that's going to be another major effort of the PanKbase community. We're going to work very closely with our clinical trialist colleagues in initiatives such as TEDdy to start to integrate data, not just from patient cells, but all the way through to clinical outputs. And I think those are some of the efforts that will really help us start to make inroads in how we can use this data in the most impactful manner.
Thank you, Anna. Any other questions before we go on to the next segment? Okay, Zoe just has a follow-up quickly, which says, thank you, Anna. And what types of cellular phenotypes do researchers need more of to answer their questions?
Oh my goodness. I don't want to speak for the entire community, but I think that being able to study different phenotypes that capture different mechanisms is really important. So clearly how much insulin you release, how much glucagon you release is kind of the end result of how your islet is functioning. But if we want to understand what might be altering hormone secretion, there are lots of intermediate steps. Cell composition, how many beta cells, delta cells, alpha cells that you have, as we alluded to in Tom's talk, is one additional metric. But I think there are other things that we can look at to do with exocytosis, and of course things to do with relating gene expression to cell function through some of the exciting new platforms that are available, including things like spatial transcriptomics. Tom, again, I don't want to be the only person giving thoughts here. Is there anything that you, from your perspective, you would add on your wish list of types of cellular phenotype that you would love to see in PanKbase?
Yeah, I think you covered it well. For as an example, PanKbase itself is working to integrate transcriptomic data and epigenomic data as well. And so I think that definitely there's opportunity in the future to start integrating the what we see in the functional data with the molecular data, both the transcriptomic and the epigenomic level. And obviously, the data I showed today, that was mainly looking at cellular composition, but also we might think about the spatial relationships between different cell types and how the islets are actually organized rather than just the composition. So yeah.
Great. Thank you. Any other questions? All right. I think we'll be handing it over to Ringo for the next section of the webinar.
All right, thank you. Let me share my screen. Hi everyone, it's Ringo from University of Michigan. Today I'm going to talk about some recent updates about PanKgraph human language search function. So first, what is PanKgraph? PanKgraph is the knowledge graph and agentic AI part of PanKbase that we build a knowledge graph for type 1 diabetes and pancreas biology. So essentially, a knowledge graph is consistent of nodes and edges. Nodes are regular bioentities like genes, genes variants, and also edge is the relationships that link them together, and they also got their own property and annotations. So using a knowledge graph, we can link different resources together, compare with a traditional way to represent data, the tabular database. We can curate everything together with specific link and store all the data into one unified database. So, for example, as a human, I can by glance see which gene connects with which variant. And for AI and computer science part, you don't need to worry about LLM to hallucinate about some links that don't exist because we already curate all the links inside the knowledge graph. Also, with the knowledge graph, we can link different resources and providing provenance. So we already integrate a lot of data across her. For example, we have integrated some pancreas atlas and hub map data, and also we are working on to include CMAD CDE into PanKgraph. And here is one example. That's the gene ADCY3's notation inside PanKgraph. So we have annotations from ensemble, HubMap, PanKbase data, gene ontology, and also PubMed literatures that all connect to the gene ADCY3. So in this way, you can start with the gene and explore all the connected resources all in PanKgraph. And the fair principle and rigor is very important for PanKgraph. We want to make PanKgraph fundable with our human language search function. And PanKgraph is accessible from both very human friendly interface and also API for computer science part. And also we use public identifier and public ontology to make sure PanKgraph can interoperate with other resources. And we use traceable and version evidence to make sure PanKgraph data is reusable. So the core idea is that PanKgraph connects resources. It does not replace them. And with proper provenance and version control, we can make sure every statement comes from evidence. And here is a example that's our interface. So you can see that we always have data source section that shows where this data comes from. And this is the current scope of PanKgraph. It includes over 5 million nodes and over 7 million edges, and it's keep growing. It covers the basic knowledge in type 1 diabetes and islet biology. We use our PanKbase single-cell RNA-seq and single-nucleotide ATAC-seq and integrate them into as knowledge into PanKgraph. And also we include fine map QTL, GWAS, and localization data, and other like HPAP metadata, and also pathway, and other annotations. And we include all the Hern literature full text, if available, and selected PubMed literature to enrich the PanKgraph. And we also work on to link more resources within and outside PanKbase. And we also have our full data dump available with Proto-OKN. And from a human language question to a grounded answer, we use agentic AI to give a solid answer. So user can just ask a plain language question, and our agent will identify relevant resources. And we use our own post-trained self-hosted model to find the right resources and evidence for you. So it's not a general LLM. It have less association, and it knows everything inside PanKgraph. So this model can help you to navigate through the PanKgraph. And we will retrieve the necessary information and answer with all the evidence and provenance. So it's grounded by design and also bounded by evidence. So it's still limited. For example, PanKgraph have no like medical suggestions. So if you ask like what's a medicine you want to recommend, then PanKgraph agent will not answer because there is no evidence in PanKgraph for that part. And we are still a relatively new initiative, and we are still keep expanding. And here is our very user-friendly UI. You can go to PanKbase and use the first tab PanKgraph, or go to pankgraph.org and just type a human language question, a plain language question you like, and hit a search. And then you will go to our plan page where we will show a interpreted version of your question and some preliminary data, so you know what data behind the we found inside PanKgraph, and also our plan to how to get data for your question. And if you are not happy with that, you can refine the plan here directly, or give feedback here. And if everything is okay, and then you can run these steps, and our agent will gather all the evidence for you. And in our result page, we have this AI summary that can explain those hard concepts for you. So even you are not expert in localization, but our AI summary will explain it to you. And also we have follow-up question. If you are not familiar with one concept, you can always follow up. And you can hover you can always check the raw evidencing table and download them, or hover anywhere in this knowledge graph to inspect the full records. For example, here is our gene detecting edge. It includes all the necessary statistics and also how we transform data into this edge, and also data version and source, so that you know where those data knowledge comes from. And also we have this literature summary section, and all those links are real. You can click and go to the resources, and in this way, we want to help everyone to make T1D researcher easier. So PanKgraph lowers the barrier from biological question to testable next steps, make all the evidence accessible to researchers, and enabling rigorous grounded AI assisted science. Thank you. That's all for my part. And if we have time, maybe I can do a live demo too.
Yeah, we have some time. So if you want to do that, please feel free. And then just a reminder to everyone, if you have any questions for Ringo as he's going through it, we can do that as well. So I think that's perfect.
Sure. Yeah, let's do some live demo. So uh, first, you can go to pangraph.org, and now you can see that we have all, you don't need to worry about like database, schema, or SQL. We have everything right in plain language. For example, all of our examples are simple questions. You can choose whatever you want or type your own question here. For example, I want to learn about if this gene is a 20 effector gene or key marker gene. So I just click search, and our agent will try to understand this question and find related evidence and query graph for that. And it's a quite complex process, so it do take some time, around like 10 to 20, 10 to 20 seconds. And here is our preliminary result. You can start to play with this knowledge graph. So you can hover to see all the nice annotations we collected. And here is the question plan. This one looks perfect. Then I just run these steps. And after some loading, our AI summary will show up all the evidence for you. And oh yeah, it's really fast. So here is all the necessary summary. We found one effector gene record, and here is all the source. You can go full screen or download this data. And we also have, oh, this string is also differentially expressed in QND versus ND case, and it's also detected across multiple cell types. Wow, it's like actually really universal, although not very high percentage of cell expression, but we do see it's like consistently expressed across the pancreas. And oh, there is no direct current literature related to this question. It's okay, because we always make sure that we only provide real citations, real reference for you. And also, like if you are interested in other part, for example, we have this two page where you can explore QTL or GWAS faster, or we also have a basic functional data tool for quick glance. You can just play with those filters, and here by one click you can ask our agent to interpret those functional data for you. And functional data are quite complex, so I have this pre-run result for you. So for example, for those not very familiar with this field, our AI summary really breaks down every section for you. For example, it tells you that the G5.6, the basal here, means that it's expected basal insulin, basal glucose stimulation. So it's just some basal insulin output. And the G16.7 is a high glucose stimulation. And we got the result. And also, for example, this one, IMBX. If you are not familiar with this, you don't know what it is. But our agent interprets that for you. So IMBX is a PDE inhibitor, which will increase CAMP production. And the result is that we expect to have some insulin secretion increase. And yes, indeed, we got the highest peak here. So you can really see how our AI summary workflow make everything more accessible, and make sure our AI always shows the necessary raw data and evidence for you, so that it's a grounded AI, not a general AI. And we always make sure we always show our provenance, so that everything is really easy to find and with solid evidence. So yeah, I think that's all for my live demo. Is there any question?
Just a reminder, please feel free to use the chat, the Q&A, or you can raise your hands and we can take you off of mute.
Thanks, Ringo. That was super. And I noted at the bottom on PanKgraph, you have a place for people to provide feedback. I don't know if you just want to show that again. So again, all of you out there, the best is to kind of go away and to explore the websites yourself, try out the tools, and just like I said earlier, we really want your feedback. So if there are things that are broken, not quite working for you, things you would like to see or like to ask, please use those feedback options to provide us with that information so that we can reflect on it and implement and bring about change.
Thank you, Anna. Also, if we're still waiting for questions, oh, and Amanda also just put in as well. And for feedback, for PanKgraph, this also helps train the agent. So it's important to get feedback from the community because it'll only make it stronger as we get more information. Because we have a few minutes left, there is one thing that I think we're excited to tease for the next stage of PanKbase. So I might walk through some of that just because we have a few minutes left, but I'll also monitor some of the Q&A. But we're really excited that we are working on a cell state and program explorer. So this is still in dev, but I just wanted to showcase what we're working on right now, which will be hopefully out soon, so you guys can see and play around with it. But this, like I said, it's just some screenshots of everything. But what this interface will allow you to do is you can start with a gene of interest, specifically one that if you're interested, you can type it into the search function here, or you can select from the options here. I should, before I get ahead of myself, also state that the importance of this is you can explore where this gene that you're searching is expressed, and then the cell states and the gene programs associated with the expression. So once you are able to put in the gene that you're interested in, you're going to be prompted to select a tissue, which you can see here. I've selected pancreas, and after that, you can select cell type, so you can explore the expression, and based off of the expression, you can select which is of interest to you. I'm having ductal here. And so once that is there, it'll prompt you to ask about the cell states and see what you want to explore. So, or the gene programs associated and both. So here I'm going to start with the ductal epithelial identity. And as you can see, if you hover over the different rows in the tables here, you get the metadata associated with it. So if you're curious and want to look into these a little bit more, this will pop up. And then again, the same thing will happen for gene programs. And then as this says, the full metadata, you can click on the different items in here as well to explore a little bit more. And as I said, then that gives you the option to finally explore the cell state and program details, relationships, and trait associations. So it first will prompt you with an overview of the inferred program. And then you can filter through the different tabs on here to see the gene loadings. And sorry, yeah, as you can see, some of these are quite long. The gene loadings, so this has the top 30 of 4,000 that you can explore here. The state matches, and then the traits, gene sets, and QC signatures. And so I did go through this pretty quickly because this is something that we are in development for. And as we've been saying for any of the items, please play around with it. Please give us feedback. If there's anything specific, this is for feedback for the gene programs and cell states. So if you're interested, please feel free to scan the QR code for that as well. But with all of the things that we've been talking about here, we want to design all of these features with the community to best help. And so we're really excited to have all of you, participate in that. So I know I went through that really quickly, but I do see we have a question in the Q&A, so I want to make sure we get to this. Zoe has said, thank you. This is all amazing to hear about. I'd like to follow up with Ringo. So Zoe sent some information on Ringo, and I can share that with you if you can't see this already. So hopefully that you guys can connect and talk a little bit more about PanKgraph. Any other questions or comments? Sorry, I didn't mean to interrupt you, Ringo.
Oh no, it's okay. Yeah, I just want to say to Zoe, thank you for your interest. Yeah, I will definitely connect with you. Yeah.
Great.
Smashing. And just to say, around the cell states and programs, this is an area, again, where we want to work very closely with the community. Some of the people on the call might be a member of the NIDDK-funded Human Islet Research Network, HERN, and we will be at that meeting next week. And we hope to engage with the community around aspects of that development. Amanda, I see you've just come on. Did you want to comment on that or something else?
Yeah.
Anything?
Yeah, absolutely. Exactly like you're saying. Hi, I'm Amanda. I'm Outreach and Engagement co-lead with Mackenzie for PanKbase. So we're glad to have you here. But tagging on to what Anna said, for the feedback, visit us at HERN, but also the link that Mackenzie had put up for the cell state. It really is to get interest in people who want to beta test for us. So while it may not be up on the site or available yet, if you fill out that interest form and let us know that you're willing to help go through this site and give us feedback and make it better, that would be great. So that's all I wanted to say.
Yes, thank you. I'll put this link in the chat as well. So if anyone wants to just have it just in case. So please feel free if you have any questions, let me or Amanda know, and we can help you with all of that. But again, to reiterate, we really want to build all of these resources with the community focus in mind. So yeah. Great. So I think that's everything that we have today from the PanKbase team. But speakers, I'll give you one more second if there's anything else you want to add. But I just want to say thank you so much for showcasing all the hard work that you've been putting into this resource and to learn a little bit more about how we build these resources. So thank you so much.
Thanks, everyone.
Thank you.
Thank you all. Have a great day. Bye.